I've really enjoyed the new food. This is new to me. It also might work as a prop later. So I think the first day, which was performed by the students and young scientists, provided such a good education for the genetically engineered 101 session. We would have gained a lot of knowledge of where to go in cancer immunotherapy. So I'm going to break the rule a little bit and just treat this like a workshop and show you what we're doing, and perhaps offer some solutions and some help to the hub so that we may more effectively form some collaborations. We are based in the bottom of the South Island here in Dunedin. Otago University was founded in 1869, so it's not quite as old as your beautiful temples around Bangkok you took me to. I was born up here in Christchurch. This is the old university building here, the registry where the managers sit. And this is our current lab group here, and present a lot of data from just a few people. So we're happy to have a collaboration to Thailand, and I hope the arrow goes to the right place this time. It certainly does to Dunedin, and I think this draws together some expertise across the Pacific. Hopefully you're going to strengthen areas here in Indonesia, Malaysia, and as well as in Thailand and the surrounds. And one thing we have done is we have shared a student, Asmita, who is a PhD student in Chulalongkorn, who's currently finishing in vivo experiments in our laboratory. So I just talk about CAR improvement, and some of these are quite conservative changes you can do to non-coding regions such as promoters, and also 3' regions, which I'm not going to talk so much about. Exploiting bidirectional promoters to get really good expression in 2 directions to save some space if you're concerned about lentivirus cargoes, and making sure you can maximize the payload that you put into the T cells. Auto-inducible promoters for payload and self-driving cars, and expanding normally discarded NK cells from the CAR T cell protocol. What do we do with the things we throw away? We like to recycle in New Zealand. I'm sure you do in Thailand. These are NK cells covered in beads, dextran beads. If you ever looked under the microscope, you see they're absolutely coated in beads. We can recover them and expand them quite nicely. In fact, we get more expansion with NKs than we do with the CAR T cells. So 27 slides only, so you can count them down. I'm on to the 4th one, so I'm about 15% done. If I go over time, you will tell me off, I'm sure. So first aspect is bidirectional promoters, which we're working on with Natia and colleagues. Fire and Coromit also involved with this project, driving it. And if you've got a promoter, you might just think a promoter like EF1, elongation factor 1, is really good for driving a single gene product. Well, it's actually great for driving 2 gene products. You don't even have to put a Tata box in the reverse direction. It seems to work without that. And of course, we discovered this by accident. So this shows you can put a gene of interest on the reverse strand, have one up here going forward, and you get really nice expression, which I'll show you in a minute. We've investigated that for quite a number of promoters that surprisingly have strong bidirectional activity. Where this might be relevant to CAR T cells that we're investigating with our Thailand collaborators is that if you lose the expression of CD19 from leukemic cells, still got hopefully CD20, and you can have a dual CAR system to target those. It's exactly what we're working on with the Thailand collaborators. So I'll just give an example. This is a small promoter called RPBSA. So it's derived from a housekeeping gene that has a strong promoter. And by placing RFP in the reverse direction, GFP in the forward direction, you can see it's got very strong expression, and you can even reduce the size of these promoters down to quite modest size, 430 base pairs, and you still see strong activity in both directions. That's just one example. We've taken this to a number of other promoters, looked for the activity. Our favourite is EF1. So the good old EF1 will work really nicely in both directions. So lesson 1 is you can save a bit of space in lentiviral vectors by doing this, or in sleeping beauty systems by expressing from a bidirectional promoter. This study's turned out well, but I'm not going to tell you the results because I'm going to leave that to Asmita, who's just finishing a PhD, And as we say in English, I don't want to steal her thunder, so that she will have the opportunity to present that. Again, Ali Hosseini, Rad, was a postdoc in our lab, came here to Thailand and started all this collaboration up. So thanks Ali. So I'll move on to the second point, which is payload delivery using auto-inducible promoters. And if you want to target tumours in a specific way, it's nice to have the CAR T cells sensing that there is something there and then releasing the payload, which might be granzyme and perforin, or it might be some cytokines to excite the tumour to be hotter. So designing these, we generally in our laboratory like to optimise things using lots of postdoc time and PhD student time up. They get quite frustrated, but it's really necessary because if you look at the designs of lentivirus, you can really go wrong. If you tried this one, for example, this design, you'd see that your titers are just miserable. You'd end up in tears. So it's really important if you're trying something new in lentivirus to try everything, every direction. So every direction has been tried here by Joshua Halpin, and these are acceptable titers for our lentivirus constructs, even this one here. Poly A was included because it increases protein expression, but it seems to interfere with titers. So using our favourite auto-inducible promoter, which I'll now reveal is NR4A1. I didn't reveal that in July, but we're close to publishing it, and I don't think we can patent this, so there you go. Payload delivery there. A forward direction where both genes are facing forward or one is facing backwards. This one seems to be the better one. Using a poly A will drop your titers of your lentivirus, and it gives you quite good expression, but we would choose this setting here, a reverse direction for the inducible payload. This is DS red. You can put cytokines there. You can put anything you want to make the tumour hotter. You can put things there to make the T cell move towards the tumours, as was suggested by John Mayer. And this is an inducible fashion, so it keeps things quiet outside the tumour. Systemically, things are quiet, but at the tumour site, things are getting hot because the CAR T cells are releasing in an inducible fashion. One simple thing you can do with inducible promoters is drive the CAR itself. So these promoters, these inducible promoters, they're not like the tetracycline activator in TN10 of E. coli. They're leaky. They're inherently leaky. Anything in humans is always a little bit leaky. No matter how you try to make a tight inducible promoter, you probably can't. So there is always a little bit of basal expression of CAR, and that's the trigger for getting more CAR. So that you're moving from a little bit of CAR on the surface to getting more CAR from an inducible promoter. So we've come a long way over the years, and we've settled on we settled this year on about 3 promoter designs, the auto-inducible. There's lots we threw away I'm not going to talk about, and we looked at these NR4A repeats, which are the target repeats recognised by a series of proteins called in the NR4A family that are induced during T cell activation. We looked at the NR4A promoter itself, which is our favourite one, which is the one we're taking further, and that's just the native 360 base pairs of upstream minimal promoter of this region here. There's just a very well characterised NFAT, NF-kappa B repeat there using a minimal IL-2 promoter, and these were compared for inducing the CAR itself. So the CAR is linked to GFP. You can see the GFP expression there on EF1 is there when it's induced, and when it's not induced, no surprises there. But the NFAT, NR4A, and this repeat, you should see more induction of GFP and both CAR as you activate the cells, in this case with CD3, CD28. So this is using a real antigen, a real tumour antigen, HER2-FC bound to a plate. You can see that these 2 inducible promoters here have fairly good fold induction, but this NR4A repeat there, we've thrown that away. That was published by Go et al. And was reported to be fairly good for CAR T cell use, but didn't really operate that well in our hands, so we've discarded that one. Now I'm going to go straight to tumour experiments, and then we can perhaps have a run through why we got these results. So this is EF1, a very strong promoter driving the car. MCF7 is the tumour model. We're putting in 2 million tumour cells, and then 4 days later, we're putting in the car. So this has cleared most of the tumours in the mice, one remaining. NFAT was doing okay. We lost a mouse there, and NR4A1 was the best out of that. You can see the PBS mice losing mice quite quickly in those groups there. And if you look at the luminescence or flux, sorry, graph there, you'll see that the EF1's up there, and the NR4A1 is actually performing better than good old EF1, which is in the FDA-approved CAR T cells. So why is that? If you look at the promoter designs, you can see EF1 is really enriched in the core promoter elements, which, you know, kind of like the engine, and there's upstream elements there of transcription factor sites. EF1 is very strong. It's got the most number of transcription factor sites overall. And these other ones here don't differ that much between them in terms of core promoter elements and in terms of transcription factor sites. Now, I've done this transcription factor site comparison, is just putting them all together and seeing who's got the most of each site that are relevant in T cells, something we did for our promoter comparison paper when we looked at constitutive elements in CAR T cells. And you can just see that there's some strange differences there. So STAT5 sites are enriched in the NR4A1, and these ATF sites also enriched there. But really, if you look at that figure, you'll see there's just a mass of blue. EF1's it's a longer promoter. It's got many transcription factor sites, but surprisingly, it's not doing as well. And you're probably thinking that it's either cytotoxicity or exhaustion. There's some difference there. Cytotoxicity here, 2-to-1 effector ratio, so not a lot of CAR T cells there. No difference between those constructs there, untransduced and empty there, and killing MCF7 with HER2. In vivo, we took out the cells. We saw no real difference with PD1, but with TIM3, there appeared to be higher TIM3 expression on EF1 and lowest on NR4A1, so exhaustion might be part of the explanation. PD1, again, we didn't really see a difference there. You can see the results summarised there. So not a major difference there, and not a consistent difference for PD1. If you do an in vitro stress test with them, what you notice is that with the inducible promoters, you get less of these effector memory cells there in that part of the quadrant there, and more with EF1. So there's some evidence there's more exhaustion happening on the T cells with that setting. And you can see here that these T effector memories, which are probably the more exhausted ones, are more numerous in the EF1 compared to the two inducible promoters, but not a lot of differences there. Nothing I'd really say for sure why there was a difference in the mouse experiments. If you do the in vitro stress test, it can also reveal that there's appears to be some tonic signaling with EF1 driven CAR. There's a basal level of PD1 expression that's definitely higher than the two inducible promoters. But this one here drove the CAR that behaved the best in the mouse model. Okay, so finally I just pause and we can talk about expanding and engineering NK cells for immunotherapy. And I hope I can convince you that despite some unhappiness around NK cells in immunotherapy not perhaps living up to their promise, there is a place for them. It's been emphasised widely that NK cells do not induce GVHD like T cells will, and there are natural or multiple natural pathways for destroying cancer cells, especially the NKG2D ligands that are upregulated with stress on tumour cells, and as we saw with John's talk, also on the surrounding stromal cells. They can participate in conjunction with antibody to induce ADCC. So if they've got CD16, molecule. They can lyse tumour cells through therapeutic antibody, and they can also be engineered with CAR. But what I want to emphasise in the last part of the talk is the promise of combining these with CAR T cells in a single patient product, the possibility of this. So if you want to expand NK cells, I would highly recommend you use our system. All of these constructs are deposited in Adgene. They are all Sleeping Beauty based, and the good thing about using Sleeping Beauty system developed by Eric Kovarts is that these are really good cassette contexts for putting a gene in and making sure you get high expression. If this was transfection, you would not see those levels of expression of all those transgenes, and I haven't even shown IL-21, membrane-bound IL-21. So those are all, it says coming soon. They're actually in the Mclellan lab gene collection, or perhaps we can collaborate if you want any iterations of those. And they're published in this paper in Immunology and Cell Biology, Sleeping Beauty kit set. So they give you a nice antigen presenting cells to expand NK cells. So these are artificial antigen presenting cells, and we use the K562 leukemic line as a basis. It lacks MHC1. It has a lot of endogenous ligands such as CD80 already in it, and adhesion molecules which will help stimulate the NK cells. So this is the final optimized setting, human NK cell expansion with these AAPC. You start with relatively low percent of NK cells, might be about 5 to 8%. And even after day 7 with the AAPC, a single shot of AAPC, no exogenous cytokines just yet, you will see that the NK cells have already jumped up to 85%. You can add IL-2 at the stage and you can keep expanding them and you get a lot of NK cells. And now we're up to about 450-fold expansion there over 14 days. We generally stop before 14 days because we run out of cell culture media, we bankrupt the lab. We're throwing cells away. So they're cytotoxic. This is two breast cancer lines showing that they will kill solid tumour lines. And we can also transduce them quite effectively with a CAR. We were surprised how well this worked actually with those activated NK cells. This is PCCL-SYN, which hasn't got a CAR in, it's just a control. So they really take up lentivirus really nicely. And this is just a VSVG envelope system. We've tried the baboon retrovirus system, didn't really improve things for us. But it certainly took up quite a lot of this HER2 CAR. And this one here, the Bayvillus one, which is seen advertised in the literature as being better than VSVG, didn't really do it for us. So I just want to wind up with the possibility of future directions, certainly for us, and certainly in collaboration with Chulalongkorn University, will be combining NK cells and CAR T cells together in therapy. And I'll just explain why, because if you're taking, just for an experiment, imagine this for an NSG experiment, you're taking 50 mils of blood, approximately 50 million PBMCs, and you subject them to bead sorting, which is what we do just to isolate the T cells. We throw away the non-T cells. So this is the T cell fraction here. We end up with perhaps a modest amount of CAR T cells there, factoring in cell losses during the bead selection. If we're taking the beaded NK cells, so that's the orange surrounded by these dextran beads, absolutely covered in those dextran beads. If we culture them with AAPC at a really modest ratio, don't need too many AAPC, they'll shed their beads, they'll continue to divide, you can recover them from that setting. So it's nice and easy, you don't have to change bead separation systems. And these expand like crazy. So it's really, would make me angry to throw those in the bin and lose the potential of isolating a lot of NK cells to use in combination therapy. You could inject them before the CAR T cells, make the tumour hot, and or you can inject them at the same time, and or perhaps even afterwards, and you can also engineer them with a CAR. So that's where we're heading. And I'm going to finish hopefully early, so we've got some time for questions or for visits to the GMP facility, which is exciting me. So these are the people that did the work here. Ali Hosseini was a PhD student, instrumental in setting up a lot of these assays with the lentivirus absolutely brilliant scientist, did a postdoc with me, did a postdoc with Ngāti Whai and Coromit, and then went to Kite Farmer. So you don't get much better than that. I think it was Ngātia's training that made him go to Kite Farmer, not mine. Asmita is finishing a PhD, one of Chulalongkorn's students finishing PhD with us, and external CAR T cells. You might not be aware, but Wellington, which is in the North Island of New Zealand, is running CAR T cell trials using a CD28 TLR2 based CAR, and getting nice results with that. No neurotoxicity, even at quite high doses of CAR. Mike Kershaw and Phil Darcy, Peter Mack have been absolutely wonderful to us. So if you're ever in Melbourne, these are great. It's a great centre to visit if you set up collaborations. And of course, our wonderful hosts and Chulalongkorn University. And remember that the world's not flat, as in one of those first slides. It's round. It takes a little bit of time to get from New Zealand to Thailand, 24 hours for me, but really pleased to have the collaboration going. Thank you so much for your help in getting me here and hosting me. It's been wonderful, and you've been a tireless host. So thank you.